Part 3. Virtual Screening, Denovo Design, Improving Lead Molecules and Examples. Now, docking can be used as an approach to virtual screening. In normal screening of potential drug leads, what we do is try basically thousands of different small drug molecules or fragments of drug molecules and screen them against some sort of biological assay. In virtual screening, we're using a docking approach to look at thousands of potential drugs and see whether they will dock with the protein of interest. So that can be used to scan a library of potential molecules and to identify drug leads. So I'm not really going to say any more about that because I think the applications are fairly obvious. The techniques are exactly as we've described. Generally, we're probably going to be using a single low energy conformation of the small molecules because of speed requirements, and initially we're probably just going to be using a very simple complementarity type scoring so that we can screen huge libraries either of fragments or of whole potential drug molecules. Once we have done an initial filtering, we might then go on to do some more detailed evaluation of those, perhaps using flexibility of the ligands and perhaps using a fuller potential for scoring the orientations. But what we end up with, whether it's virtual screening or proper screening in vitro or in vivo, is drug leads. These are not likely to be actual final drug molecules, but they are things that we can then go on to try to refine and make ourselves a better drug, something that will stick better and will be more specific and have the characteristics of a typical drug. So before I talk about how we can improve lead molecules, I'm going to mention the idea of de novo drug design, where instead of starting with some pre-existing molecule, we try to assemble a molecule from scratch. So again, there are a number of programs that can be used for doing this, and they rely on starting by putting small fragments, and a fragment might just be as small as a hydroxyl oxygen, or it might be a phenyl ring, or any other small kind of fragment that you can think of, and seeing where those might fit into a pocket. So LUDI, which is part of the Insight molecular graphics system, allows you to screen fragments that can bind. And GRID and Excite are two programs that use some sort of potential to find interaction sites for probe groups. So GRID uses a full kind of molecular mechanics type potential and uses very, very small fragments, generally, as I said, something like a hydroxyl oxygen or carbonyl oxygen and that sort of thing, and finds where those would like to be on the surface of the molecule. Now, Excite is very similar, but instead of using a molecular mechanics potential, it uses an empirical potential derived from looking at protein structures and looking at interactions of groups within protein structures. Once you have those probe groups and where they would like to be, you need to be able to connect them together into some sort of complete molecule. Again, there are a number of programs that can achieve that for you. One of them used to be called Lego because it's the idea of assembling little building blocks. I don't think it has that name anymore because I imagine they got approached by lawyers representing a well-known Danish toy company. There are also various pieces of software that allow you to drag and drop little molecular fragments and draw a picture of your molecule, and these programs will also put together a kind of low energy assembly of those atoms so that you can use that as a starting point for doing your docking. So if we bring together then the ideas of docking small ligands into binding sites, together with these sorts of analyses of where particular groups would like to be, things like grid and excite, then we can start to enhance the initial ligands to improve their binding and improve their specificity. So what we see on the left here is an image from the latest version of grid, and what we're seeing with these little kind of clouds is positions where particular probe groups would like to be. So in yellow we have sites where an aromatic carbon would like to be, and in red we have sites where a carbonyl oxygen, which is going to be an acceptor of hydrogen bonding, where those would like to be. So you can see that this molecule here, which is a MAP kinase inhibitor called SB4, you can see that here we have a nice yellow patch where aromatic carbons would like to be, and indeed this fits really well with where this kind of phenyl group is sitting. Up the top we have an interaction with a backbone atom here, and we have a site where a carbonyl acceptor would like to be. In fact, we actually have a nitrogen acceptor rather than a carbonyl, but at least it's clearly accepting a hydrogen bond here. And over here we have a site where we would like to have an aromatic carbon, but currently we don't. So on the right we have a modified version of that inhibitor. This one's now called 6NP, and that has added a fairly rigid ring structure here and another phenyl group here, such that that is ending up in that yellow patch. And this inhibitor is 7 times more potent than this one, and so this is the sort of thing that we can start to do by modifying these structures to put groups where we want them, using a relatively rigid structure in order to minimize the entropy loss on binding and maximize the Gibbs free energy, the delta G. Now let's look at a couple of examples of drugs that have been designed using this sort of approach. So the first really designed drug, designed from scratch, is the molecule known as Relenza. Its non-commercial name is zanamivir, and it's used for influenza. They used grid in order to do this. They looked at neuraminidase and identified sites on neuraminidase where small probe groups would fit, and then designed a molecule that satisfied those interactions. Another example is ritonavir and indinavir. These are both drugs used to treat HIV, and they were designed to inhibit the HIV aspartic protease, which we saw earlier in previous examples. So here again you can see the HIV protease, a nice pocket in here, and this is the molecule that was designed to fit in that pocket and block it very specifically. Now that's all very well, but we have talked, particularly at the beginning, about side effects and the idea that a lot of small molecule drugs are not very specific. Now, there are different types of side effects. So there are on-target side effects. That means that your drug is interacting with the protein of interest and it is probably having the desired effect, but it's also causing other effects that perhaps you don't want to happen, because by inhibiting that protein or blocking that receptor or activating that receptor, as well as the good things that are happening, we're also getting bad things happening. The other type of side effect is the off-target side effect, and that's where the lack of specificity of a drug comes in. So in other words, the drug is interacting with what you want it to interact with, but it's also interacting with other things, and that causes the problem. However, in some cases, this can actually be a positive benefit. So some years ago, there was a drug that was designed for heart problems, specifically for angina, which had some unexpected and interesting side effects. So a lot of the patients that this was given to during trials were men in their 50s and 60s, and it turned out that it really didn't help with the angina at all. Now, these side effects ended up making the drug company Pfizer an awful lot of money, because this drug ended up being sildenafil or Viagra. Now, from a pharmacological point of view of what this was trying to do, the aim of sildenafil was to open up blood vessels to relieve the angina, but in fact it ended up interacting with a related protein that opened up blood vessels in different parts of the body. In summary, what we do for drug design then is to find pockets, and then we need to do docking. So we looked at the principles of docking, in particular the importance of complementarity of the surfaces. We looked at docking, comparing the rigid body idea with ligand flexibility. So in some cases, software can only do rigid body, but of course can also generate, or we can generate, multiple different conformations, each of which can be docked in a rigid manner, or the software like Gold, which can incorporate the ligand flexibility as part of the docking process. And we then generate lots and lots of possible conformations of the ligand and possible orientations of the ligand, and we can then analyze those in order to find the most likely actual docked conformations. We also talked about virtual screening, which is the idea of using this sort of approach to screen a large number of molecules or fragments of molecules, which you can then modify and enhance, as we saw with that example of a MAP kinase inhibitor, or you can use those fragments in order to assemble a complete ligand. And the approaches used for doing that sort of thing are largely the ideas of things like grid and excite, which are used to identify probe interaction sites, and that can be used for those two jobs of enhancing existing ligands, as we saw with that example, or building ligands de novo.